Thank you very much for the kind introduction. I also want to thank Vadim and the Center for Physical Genomics and Engineering for organizing such a wonderful symposium. I have learned a lot from the previous two talks. And I'm sorry I cannot be there between meetings, but I hope this will work out. So today I want to talk about some of recent studies on mechano-epigenetic engineering. The research in our group is focused on cell and tissue engineering. And on the one hand, we are interested in understanding how biophysical factors in the microenvironment, such as mechanical factors, electrical signals, together with biochemical signals and biological molecules to regulate cell functions. And on the other hand, we are interested in integrating what we learn together with materials engineering, drug delivery, and stem cell engineering, immunoengineering, and use that knowledge for in vivo tissue regeneration and disease therapy. So today I want to just focus on our recent work in vitro to engineer cells by epigenetic modulation. I was trained as a mechanical engineer, and we all know that in the body, at different levels, from systems to organ, tissue, cell, and molecular level, mechanical forces exist everywhere. And our focus is at a cellular level, because for tissue engineering, if you can engineer the cell, which is the building block of tissue and organ, then we can reconstruct the tissue and organ together with extracellular matrix in scaffold, etc. On the other hand, we want to understand how at the molecular level, the mechanical factors can impact the cells. So half of our lab is focused on mechanobiology and mechanotransduction. In the past 20, 30 years, in a lot of work in the field, and this has been widely studied. And we all know that in the microenvironment around the cells, there are many physical factors can impact cell functions. For example, in the cardiovascular system, we know there's a fluid shear stress due to the blood flow and pressure changes, etc. And also cell adhesion substrate. There are many components here. In the tissue, you may have a dynamic mechanical stretching, and also the micro nano topography of the matrix can affect the cytoskeleton organization through the cell adhesion, and then affect the nucleus in the chromatin organization. In the past 10 years also, the stiffness or viscoelasticity of substrate has been studied as well, and a lot of interesting work in this area. In addition, even the distribution of the ligands, in the chemical ligands, could affect the cell functions. And the cells need to integrate all of this information and make decisions to migrate, differentiate, proliferate, etc. So today I want to focus on a couple of things. One is the cell adhesion related mechanotransduction, also in the cells. So in the past, most of this paradigm is on how these signals can be transmitted from the cell surface to the cell nucleus. And we know that many signaling molecules on the cell surface, ion channels, adhesion receptors, they all can serve as mechanosensors. And then through signal transduction, very similar to what could be activated by growth factors and ligand receptor binding, and these signals can be transmitted into the cell and then activate transcriptional factors and regulate gene expression. In addition, there could be a direct mechanical coupling through cytoskeleton that can have a physical impact on the signal transduction as well. So many of those reports in the literature focus on the transient effect of mechanical signals. For example, the ion channels or integrin activating of MAP kinase, etc. And many of those kind of adaptive responses It could be activated and go back to basal level. And that represent the quick responses in probably under physiological, pathological conditions that cells can sense and adapt to the environment. So what we're interested in is the cell memory change, which is released the epigenetic modulation. And this is very important when the cells have the phenotypic changes during the stem cell differentiation and cell reprogramming. So I think this doesn't need too much introduction because everyone knows about this. I just want to make 2 points. So here you can see the heterochromatin and euchromatin structure in the nucleus. In a couple of things. One is we know the regulation of all of these modifications that can control the opening and closing of the chromatin structure. You can see a bunch of enzymatic regulation that can control the histone modification or DNA modification. But we yet do not know this operating system. It's just like a computer. We know a lot of commands, specific commands that can induce certain changes. But really the principles behind this, just like Newton's law in engineering, to guide the whole system, how this works, is not clear. The other thing I want to point out is from engineering perspective, we always think, how the mechanical or physical perturbation of this could induce any changes. Right? From previous talk, you already see these some of these very, very good examples, phase separation, liquid gel transition, etc. And we are very interested in how this could be induced by extracellular factors. So besides all these enzymatic modification of the chromatin structure, and how can the mechanical factors affect the intranucleus chromatin organization? So here it shows the schematic from the reviewer. Then that shows how this could happen, and I use that to illustrate our hypothesis. So cells, they attach to each other through cell-cell junctions, or attach to extracellular matrix through integrin binding to extracellular matrix. And all of the signal mechanically can be transduced and coupled to nucleus through nuclear lamina. And one thing interesting, as you have seen in previous talks in many papers, that the heterochromatin, which is unused chromatin that generally organized in pack around the nuclear periphery. So here actually provides an opportunity for us to test how the mechanical impact from extracellular space could affect chromatin organization. So one thing possible is that when you have a cytoskeleton reorganization, that can induce some changes in the nuclear lamina, and then could have a further impact on chromatin organization. For example, the chromatin is associated with nuclear lamina through this lamina-associated domain, etc. So, and there are also a lot of enzymes distributed around the nuclear periphery, which is kind of strategic position to regulate, for example, DNA methylation, etc. So that provides a basis that the mechanical forces could have some direct impact on chromatin yet to be verified. And the other thing is at the cellular level. So we are interested in stem cell differentiation. From this Wendington landscape, you can see if we use this metaphor that the cell differentiation can be described as a ball rolling down these hills and valleys. And you can see the 2 things important. One is when the cells fully differentiate, they reach a stable state. We can use physics term to describe this. In between, during this process, all of this epigenetic state at each point is unstable. A small perturbation could affect the cell fate. So physically, we could think that during cell differentiation, probably there's a less barrier, epigenetic barrier in this case, to manipulate cell differentiation, and where versus this stable state, then you have much big barrier. So if you are interested in how physical factors could modulate the stem cell differentiation or cell reprogramming, so there are a couple of strategies. For example, we could manipulate those physical factors during cell differentiation or reprogramming process. And yet the most challenging part is to overcome this barrier. For example, directly convert fibroblasts into cardiomyocytes, neurons, and other cell types, or all the way back to a pluripotent state. So here I want to show you a couple of examples. We're trying to do is addressing this question, whether biophysical factors can work together with transcriptional factors and biochemical factors to help and facilitate these cell phenotype changes to overcome this epigenetic barrier. So here just shows in general how the cell phenotype could be manipulated, the natural and spontaneous differentiation from pluripotent cells, and go through all these pathways to specific cell type. And now we know from Yamanaka and many others' work that fibroblasts, this could be reversed and be reprogrammed to pluripotent cells using Yamanaka factors or various combination, even microRNA and chemical cocktails. And yet there's also a possibility to convert the fibroblasts into other cells, for example, neurons, by direct conversion. So I want to show you briefly one or two slides on this part. This is a previous published work, and we'll focus on this model, which was developed by Wernick's group at Stanford to directly convert fibroblasts into neuron. So the reason we use this model is that compared to stem cell differentiation, when we do a cell reprogramming experiment, in this process, in general, happens relatively quickly, especially in the first few days. And those epigenetic changes could determine the long-term fate of these cells, and then could be easily captured in an experimental system. So here I just want to show you a slide of our previous work 10 years ago, in when we try to manipulate the cell morphology, especially the nuclear morphology. So what we did was to culture the cells on micro pattern surface. In this case, parallel microgrooves in the cells show very elongated shape. And when we manipulate the microgroove size to about 10 micron wide in 10 micron spacing, in this will impact nuclear alignment and nuclear elongation as well. And at that point, we could see there's a global change of histone modifications. And here we perform single cell study. In this case, we use micro contact printing to generate individual matrix islands. And then we can control the shape of these islands. And one, for example, is a circular shape. The other one is elongated shape. And these two kind of shapes, they have the exact same area, but different shape index. So here, just show you one example. If you stain for histone 3 acetylation, H3K4 methylation, trimethylation marks, and these are all the marks for euchromatins, and we can see this increase globally, and also specifically at certain sites of promoter of pluripotent genes. We did ARCA4, SOX2, and NANOC as example. At a promoter of ARCA4, we didn't see much change. With the cheap PCR, we could see indeed the ACH3 increase dramatically at promoter of SOX2, and also the slight increase of trimethylation of H3K4, and a NANOC promoter at different of these histone marks increase. One is ACH3, and also the dimethylation of H3K4. So that actually gave us some clues that indeed the physical change of nucleus could have some impact on the histone modification, and then the potentially the accessibility changes for gene regulation. So here is a work in revision, and what we want to do is directly test if you relax the intracellular tension by the disruption of actin myosin interaction. Right? So if the hypothesis is right, the connection between the cell adhesion to nucleus is through actin cytoskeleton. If you disrupt this, what will happen? And I will just show you a few slides on some important data. For this case, we use the ABM, the 3 transcription factors identified by Wernicke's group. What we found is that during the reprogramming process, ASCL1 specifically, the increased cell adhesion, cytoskeleton organization, actually that could cause and form a barrier for reprogramming. Efficiency is low. If you do embryonic fibroblast conversion, efficiency is reasonable, but if you use adult skin fibroblasts, efficiency is 1 magnitude lower, down to single digit or even less than 1%. So what we did was to disrupt the actin-myosin interaction by using blebstatin, which is commonly used chemical interference. And this shows a dose dependency on the reprogramming efficiency to convert fibroblasts into neuron. At a low dose, you have some slight enhancement. At a 10 micromolar seems to be optimal. Of course, if you go beyond that, it could be some side effects because cytoskeleton is involved in so many different things and can affect various signaling events as well. So now the question is whether this cytoskeleton disruption directly regulate this neuronal gene expression or indirectly. So we did one experiment, and in this case, we could just test fibroblast cells without any reprogramming factors. BAM, these are 3 reprogramming factors, or with the 3 reprogramming factors. So in the absence of reprogramming factor, you just take fibroblasts and treat them with blebstatin. And for mesenchymal gene, I didn't show you here, indeed, when you disrupt cytoskeleton, there's a decrease of expression, which helps because if you want to convert mesenchymal cell types into neuron, which is epithelial type, there's this mesenchymal to epithelial transition. That's helpful, but that's through a different mechanism because it's known that some transcriptional factors related to actin polymerization, their translocation into nucleus, such as MRTF1, etc., could regulate that process. But for neuronal gene expression, these neuronal genes are turned off in the heterochromatin in fibroblasts. They are off. So when we add a bleb, and you can see here the first 2 bars, and almost nothing for these genes. So disruption of cytoskeleton alone does not induce this gene at all. Yet when you have the reprogramming factors introduced into the cell with viral constructs, and then you add this blebstatin, you can see this enhancement compared to the control with DMS. So this shows that there's some kind of a synergistic effect. The cytoskeleton is doing something, and potentially through epigenetic modulation. Then we examine a few histone marks. We also examine the DNA methylation as well. And by the way, when you disrupt the intracellular tension, the DNA methylation globally also decreases. I didn't include that data here. I just want to show you a few. ACH3, H3K4 trimethylation, H3K4 methylation. And for all of these, when you treated cells with blebstatin, they all increase. This is globally. And how does this happen? It's possible, as I said, it could be a mechanical effect. It could also be chemical enzymatic effects through all of these enzyme hat activity and etc. So in this case, when the cells in culture, when you treat cells with any chemicals, actually, I think it's there's a combination of mechanical and chemical effect because there's a chronic effect. There'll be time for the signaling events to happen through hours or even days of reactions. So indeed, when we examine all of these enzymes, hat activity increase dramatically. And on the other hand, HDAC activity decrease. These are all global nuclear extraction assays. And in addition, the HMT activity for H3K4 increase as well. And then the HDM activity, which is inhibitor of the methylation or decrease. So this all correlates so well. And whenever we observe these global changes and examine this enzyme, it seems the enzyme all change in the same direction. And now we want to see whether specifically if the hypothesis is right, the cytoskeleton disruption, it could enhance the accessibility of some of these sites in heterochromatin, for example, a neuronal gene, the promoter of neuronal gene. So we did an ATAC-seq. We generated a genome-wide map of accessibility and look into a few examples. Here, I just want to show you a few sites. For example, beta-tubulin, the neuronal marker, ASCL1 and MYT1L. And you can see the promoter or enhancer of these genes. There's a big increase at some sites, and you can see the difference. So that suggests that it is possible when you disrupt the intracellular tension, you increase the physical accessibility at certain sites, and that together with the reprogramming factors that allow them to bind to these sites in the heterochromatin to enhance cell reprogramming. Indeed, we also analyzed the ASCL1 targeting site. ASCL1 is a pioneer factor for reprogramming, could help open up the heterochromatin structure. And we found many sites are also open after you treat cells with blebstatin. So that's one part of work. We're trying to see how this intracellular tension could affect nucleus. The other thing we want to do is, in a literature, there are already some reports showing various cultural conditions, the stiffness or mechanical loading could affect the epigenetic changes. And yet there are some conflicting reports on how this could happen. Sometimes people showing increase or decrease of certain modifications. So I think in general, these microenvironmental factors, biophysical factors, is a context-dependent regulation. So when you have cell adhesion or even different cell types, the matrix adhesion in cytoskeleton, cell spreading, all of these could have impact. So what we want to do is now let's remove all of these systems, remove the cytoskeleton, everything, and just bring cells in suspension. And hopefully we can directly perturb the nucleus in a way to induce a nuclear deformation and see what happens. So hopefully that's a more clean system physically to perturb the nucleus. So in the literature, people have used various techniques to achieve that. Micropipette aspiration, you have negative pressure here to suck in a part of a cells or nucleus. And by the way, all of these technologies have been used for single cell or even single nucleus manipulation. In atomic force microscopy, you apply a compression, and then you can one is to measure the mechanical property changes, and two is also induce some changes physically in the cell nucleus or optical tweezers at a local. This is a more kind of a local manipulation. So all of this will give us some really detailed information in cells or nucleus at a single cell level or single nucleus level, yet it's a low throughput. For us, if we want to figure it out, for cell differentiation reprogramming, and also some molecular analysis, we need a large number of cells. So we decided to go for the very last technology platform is shown here, which is microfluidic system. So this system actually, there's also some flexibility to manipulate. For example, you can manipulate the sizes of channels to control the magnitude of compression or deformation of nucleus. You could also have these cyclic changes of the shape, so you can apply either constriction or relaxation to cell nucleus cells in a cyclic way. So we use this system to perform the study to test directly how nuclear deformation could affect the chromatin organization in cell reprogramming. So here it shows the experimental procedure. We first introduced all these reprogramming factors, BAM, 3 factors, and this is a DOX-inducible system. So we activate these genes, and then transcription factor can be expressed. And we have tested this actually a time course between 6 to 20 hours, and that's an optimal time to induce cell nuclear deformation. So these nuclear deformation is very, very fast. And this is very different from what we did in the cell culture, which takes hours or days to see these gradual changes in cell adaptation. Instead, here is a microsecond. I think it's even shorter. I need to just go back to. I think it's microsecond deformation of cells. A millisecond, I think, millisecond, sorry, just make sure. And we're also trying to make a high throughput device to even have a shorter nuclear deformation. In this case, the idea is to make something similar to flow cytometer. And we can have a microfluidic system applying this quick deformation of cells within minutes. You can process millions of cells and then use that for cell engineering. And then we culture them for various lengths of time to look at the cell reprogramming, maturation, etc. And there's one thing interesting and also important too for this system. When we apply the physical deformation to cells, we need to be careful. Otherwise, it could damage the cells as well. So this is a cell size dependent when you use this microfluidic system. And here it shows how we characterize the fibroblasts, even for mouse, adult mouse fibroblasts. It's heterogeneous. You can see this Gaussian distribution, cell diameter. But the most important, we care about the nuclear diameter. And here is about 10 to 11 microns in average. But there are some cells bigger and some smaller. And if you use different sizes of the channel, and by the way, for the convenience of microfabrication, we make all of this channel height consistent, which is about 12, 13 micron. And we only vary the width of these channels. So you can see here 3, 5, 7, 200, etc. We did a various characterization just to make sure we don't kill the cells. The cell viability is very important. For example, here you can see this is a Lamina AC staining with H2B GLP labeling in the cell as well. If you have a small channels, you can even cause this blabbing and disruption of nuclear membrane, etc. And what we found is 7 micron seems to be appropriate for this cell type. So which is smaller than the average. That means you do have a deformation of nucleus, and then yet you don't induce significant cell death. And here, a wide channel, we use that as control. We just let cells flow through a big channel without a compression, but they are subjected to the same fluidic exposure. So one thing we noticed that when you subject the cells to this mechanical compression, and you definitely deform the nucleus, and you can wait after experiments. You can wait for hours. You can see the cells to the surface, and the cell will take hours to spread out and get back to normal stage. And we measure the stiffness of the nucleus after 3 hours, 16, etc. And in this case, we use AFM to directly compress this cell topic. This top right at the top of nucleus site. If you do this measurement near the edge of this, you probably want to measure the cytoplasm mechanical property. And if you put on top of this, it has been shown in the literature, the major contribution of this stiffness is from the cell nucleus. And you can see there's a definitely decrease of nucleus stiffness right after mechanical squeezing, and it's gradually recovered. So I think this is consistent as what we have seen in the previous talks. For example, it could be this gel to liquid transition, and you disrupt certain structure and probably change this DNA organization. And then we examine the reprogramming efficiency. And here just shows the time course. If you look at the ASCL1 gene expression, indeed within hours, if you squeeze the cell, there's a huge increase. Of course, this is a total ASCL1 expression, including those forced expression of reprogramming factor. And then we use the ASCL1 promoter-driven GLP to directly examine the endogenous gene expression. Because what we want to achieve to reprogram cell is to activate the endogenous neuronal genes. And we indeed show it's a significant increase as well. The other question we wanted to address is whether this mechanical deformation of nucleus make this reprogramming process faster or earlier. And we use these nano probes developed by Peck Wang's group at Penn State. In this case, you could detect early mRNA expression of the gene, neuronal gene. In this case, we use TUB3 as a marker. And you can see, indeed, with mechanical stimulation, you induce this neuronal gene expression endogenous earlier. So the reprogramming process actually happens very quickly. And we found that actually the first day or two are the most important. And afterwards is reorganization and remodeling. So what happened to the epigenetic marks? We again look into these, all of these histone modification in the supplemental data. We listed, I think, over 10 different kind of histone mark stainings and DNA methylation as well. Here, what we found is one major changes in this mechanical regulation is H3K9ME3, which is the heterochromatin around the nuclear periphery. And if you look into here, control, and you can see still a lot of these clusters of DNA chromatin here, and with the squeezing, you see that they are dispersed and form this really diffuse pattern and gradually recover within 24 hours. So this probably is also the indication that some phase transition and other changes, and we yet need to look into. And also, what we think is that this probably is the time window if you want to reprogram cells, and this very similar to embryonic cells or other cells, which is kind of a primitive or a stem cell state that the chromatin yet not to form, has not formed so many this kind of a closed structure, heterochromatin structure yet. And this is also transient. We quantify this process. And now the question is, people know that the enzymatic effect on the heterochromatin or just general chromatin mark changes, histone mark changes will take time. How fast this physical induced heterochromatin mark changes, specifically H3K9 in this case, will happen, how fast will happen. So we use this FRAT probe developed by Peter Wang's group at the UCSD. In this case, it detects the binding of H3K9 methylation in HP1. We put this probe into the cell, and here shows one cell, a time course, because this takes really millisecond for cells to go through. It's too fast to observe these changes. So we slow down this process by adjusting the pressure in the microfluidic system. So we lower the pressure and allow the cells to slowly go through, but this just takes a few minutes. You can see here, red color indicates higher H3K9 trimethylation level, and green means lower. And when this cell passes through, and this shows the nucleus shape change, and the signal decrease within minutes. It's very fast compared to traditional, whatever people have reported for chemical modification. The other thing we look into is DNA methylation. So similar to what we observe for the H3K9 trimethylation, there's a decrease of this clustering of DNA methylation that indicates that there's a decrease of this closed structure, and this is also transient. So what happened in a cell? Is that everywhere increase or decrease? So we did some experiments. These are unpublished. We did high-C in ataxic in sequencing to trying to figure out what happened genome-wide globally. So it's interesting if you look into this heat map, ataxic data. We showed a duplicate for two samples. So when you mechanically deform the nucleus, about 60–70% of the sites become more open. But actually, there's about 1/3 of the sites become more closed. So it's definitely there's some physical and spatial effects. And we also found some of chromatins are overrepresented. In this case, there are more changes. So if you're looking high-C data, we need to analyze those. And here just shows a couple of examples that a specific promoter or enhancer of ASCL1 in tube 3. It shows the peak changes and accessibility changes. In general, several neuronal genes promoters all shows the increase of accessibility. And the other question we address is, how can we compare this biophysical effect with biochemical effects? So that well known, many of these chemical inhibitors that can modulate, for example, here, the BIGS is H3K9 specific methyltransferase inhibitor, and then the Cetobin is the DNA methyltransferase inhibitor. So we add them alone or in combination and compare that with physical squeezing. And here showing that if you have squeezing, and you have an increase of reprogramming efficiency, and together there's some additive effect, but not too much. And for the DNA methyltransferase inhibitor, it also shows some enhancement, and then together with mechanical squeezing, this is additive. And we also try to just have a chemical combination, because now these two changes, H3K9 trimethylation and DNA methylation, if you suppress both with chemicals, actually they reach similar level as physical. So physical squeezing of nucleus actually causes more changes, and maybe that's something we haven't examined yet. Another question is about mechanotransduction. When you deform the nucleus, in this case, all cytoskeleton have been disrupted in suspension, and what happened? So one thing we look into is the nuclear lamina, which is the structural support of nucleus. And if the hypothesis is right, then all these heterochromatin affiliated with nuclear lamina could be perturbed. And here we show that if you squeeze the cell, you see this wrinkling. And definitely you make the nucleus softer as well. And then this gradually recover. And also at the beginning, if you quantify this lamin AC at the nuclear periphery, there's a slight decrease in represents a partial disruption. So then we directly examine the interactions between the nuclear lamina and chromatin by using this M6A tracer to detect the LAD interaction with lamin B1. Here shows lamin B1 in red, and M6A tracer in green. And after mechanical squeezing, you can see some partially at the periphery of the nucleus, you decrease the direct colocalization of heterochromatin or chromatin structure with nuclear lamina. So if this nuclear laminar perturbation really plays a role in this mechanotransduction, then we directly disrupt its structure. It should work. Indeed, actually, in embryonic stem cells, people have reported that you have much less nuclear lamin compared to fully differentiated cells. In here, we use siRNA for lamin A, and you can see when you disrupt nuclear lamin A, you also see a disperse of this DNA methylation over here, very similar to mechanical squeezing. So we quantify this H3K9 trimethylation and also DNA methylation, and we found that indeed there's decrease with lamin knockdown. And the lamin knockdown also increases the reprogramming efficiency when you're trying to convert fibroblasts into neuron. And this is consistent as our hypothesis that when you disrupt the lamin matrix, you probably will increase accessibility in chromatin. So the last thing we trying to figure out is whether this is a general phenomenon. Is that cell type dependent? And we try other cell types. For example, a challenge is to convert fibroblasts, which is easily obtained from circulating blood, into neuron. So in this case, we deform macrophages in a microfluidic channel, and we perform the same experiment as in fibroblast conversion. And we show that in this case, the mechanical squeezing also increase the reprogramming efficiency. And at the same time, when you convert fibroblasts into iPSC, you see much more colonies if you subject the cells to mechanical squeezing here. And we are trying to do is make high throughput microfluidic device. This is just one design with this reading configuration, and also some other designs we're trying to do is a smallest space area. We can put as many channels as possible to process many cells in parallel. So in conclusion, what we think is that the intracellular tension, the actin cytoskeleton, which is important to connect cell adhesion to nuclear lamina, definitely plays a role. If you relax this tension, you probably make some free-floating nuclear situation, allow easier chromatin reorganization. And if you directly deform cell nucleus, as we shown in this microfluidic system, that actually has some impact because this directly affect the heterochromatin at the nuclear periphery. And we believe this finding could lead to certain applications. In this case, what we are trying to do is generate a Mechano-Epigenetic map following this nuclear deformation. And we found this is consistent. And if we identify certain sites with increased accessibility, we can use the CRISPR to design specific guideline in target those sites and demonstrate it is true when you have mechanical perturbation. And locally, you can also engineer gene activation or deactivation with CRISPR-mediated gene editing. And we are trying to make a high throughput device, hopefully could have some application for cell engineering. And with that, I want to thank the funding source, the collaborators I mentioned earlier, and also the cytoskeleton work was led by Jennifer Sato, a postdoc in the lab, in the mechanical squeezing project was led by Yang Song, another postdoc in the lab. Thank you very much.